Hello, everyone, and welcome to today's broadcast, The Persistence of Pertussis and the Prognostic Value of Bordetella Detection, presented by Dr. Karissa Culbreath, Medical Director, Infectious Disease, Tricore Reference Laboratories, and Associate Professor, Department of Pathology, University of New Mexico School of Medicine. I am Xavier Gutierrez of Labroots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by Diagnostic Molecular. For more information about our sponsor, visit www.molecular.diasorin.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Culbreath. I will now turn the presentation over to her.
Hello, I'm Karissa Culbreath, and I'm here today to speak with you about the persistence of pertussis and the prognostic value of Bordetella detection. Just a little bit about the laboratory where I work. We are Tricore Reference Laboratories located in Albuquerque, New Mexico. We are a regional hospital laboratory, a regional reference laboratory, and we provide laboratory services for three major health systems in New Mexico. In addition to the hospitals that we serve, we also provide laboratory testing for commercial physician and other smaller community hospital clients throughout the state. As a frame of reference for our infectious disease laboratory, our volume is about 850,000 to 900,000 tests performed in the laboratory each year. And this ranges from the basic laboratory tests, such as cultures, to more esoteric molecular testing. All of the samples that are received in our laboratory are delivered by courier, and we have courier services going throughout the state and bringing specimens to the laboratory 24/7, and we are running tests in the laboratory 24/7. So here today we are speaking about Bordetella pertussis and other members of the Bordetella species group. Regarding Bordetella pertussis, humans are the only host for Bordetella pertussis, which is transmitted by droplet transmission. Bordetella contains several virulence factors. Of these, the most important causing disease is the Pertussis toxin. However, there's other toxins and virulence factors that play a role in disease pathogenesis, including tracheal cytotoxin, which destroys the ciliated cells and inhibits cell regeneration, adenylate cyclase hemolysis, which induces apoptosis of cells. The filamentous hemagglutinin promotes binding to various cell types, and the fimbriae that helps to support colonization of the entire respiratory tract. These virulence factors taken together with pertussis toxin are what are able to mediate the pathogenesis of the organism in the human host. There are multiple stages of pertussis infection that we are able to see in disease. The first stage is the catarrhal stage. This is the early stage that's occurring during this initial incubation period of 7 to 10 days of incubation. At this point, what we would see clinically are non-specific symptoms mimicking influenza-like illness. These would consist of a low-grade fever, sore throat, nasal congestion, and other influenza-like illness symptoms. These symptoms are generally mild, but there is a progressive dry cough that will later become more indicative and classic in the presentation of pertussis. The next stage that we would see is the paroxysmal stage. This stage is when we would see the classic whooping cough, which gives pertussis its most commonly known name of whooping cough. We also would see post-tussive vomiting. A profuse production of mucus would occur at this stage. And then the paroxysms following coughing, they would occur at night, and they increase in frequency throughout the next 2 to 3 weeks of infection. The last stage that we would see with pertussis is the convalescent stage. And at this stage, the disease severity begins to wane. The paroxysms decrease in frequency, duration, and severity, but there remains a mild cough, and this cough can last up to 6 weeks. During the course of these stages of infection, we see the organism burden be the highest during the catarrhal stage, and then it will decrease over time through the convalescent stage. And during this time is when we're really seeing the activity of these virulence factors, in particular the Persistence toxin, when it comes to pathogenesis. So a little bit more specifically about Persistence toxin. This toxin is only expressed in Bordetella pertussis, and the toxin itself is an AB toxin. The toxin is released from Bordetella pertussis in an inactive form, and then it binds the cell membrane receptor and is taken up into the endosome, and it travels through the endoplasmic reticulum. The alpha subunit itself acts as the promoter, and then once it becomes activated, it prevents the activity of the G protein coupled receptors and interferes with intracellular communication and increases cellular concentration of cyclic AMP, and it disrupts the cellular function through its ADP ribosylating activity. There are other Bordetella species that can cause disease other than Bordetella pertussis. These species include Bordetella parapertussis, bronchoseptica, and Holmesii. All of these can produce disease, but they're all have more mild presentations in general than Bordetella pertussis. Bordetella parapertussis lacks the production of the toxin due to a mutation in the promoter region. It can cause a pertussis-like illness, but as I stated before, this illness is more mild in presentation in general than with Bordetella pertussis. There are 2 hosts for Bordetella parapertussis, humans and sheep. Bordetella bronchoseptica also lacks production of the Bordetella pertussis toxin. It can cause mild respiratory illness, but not necessarily pertussis-like illness, and you may not see as long of a duration of coughing, won't see any of the paroxysms or the pertussis post-tussive vomiting that you would see with Bordetella pertussis, and then there are also multiple hosts, humans as well as other animal hosts. Bordetella Holmesii is an interesting and emerging pathogen that we are beginning to see as we're expanding our diagnostic capabilities for Bordetella species. It causes a mild respiratory illness in immunocompetent hosts, but we'll speak a little bit further about the illnesses that it may be able to cause, and we're starting to see a bit more of in immunocompromised hosts. And again, as with Bordetella pertussis, the only known hosts for Bordetella Holmesii are humans as hosts. So when we look at the clinical presentation, though, of Bordetella parapertussis compared to Bordetella pertussis, we can see that there are some significant differences, but there are some overlaps when it comes to the clinical presentation of these 2 pathogens. What we do see for Bordetella parapertussis is that there tends to be a shortened cough duration compared to Bordetella pertussis. Bordetella parapertussis has an average of 14 days, the median of coughs, compared to 28-day average cough duration for Bordetella parapertussis. The presentation of the symptoms, though, of the pertussis-like symptoms actually tend to be fairly similar, although we don't see as much of the paroxysmal cough or the post-tussive vomiting. The whoop can be present in Bordetella parapertussis. Apnea is present with a less common frequency. However, there are some sleep disturbances, fewer hospitalizations, and then fewer deaths are also seen with parapertussis. And what we do see, though, is that many of the patients who would have Bordetella parapertussis are up to date with their vaccinations for pertussis, and that's because there's no protection, no crossing protection between Bordetella pertussis and parapertussis when it comes to vaccination for Bordetella pertussis. When it comes to the presentation for Bordetella Holmesii, what we do see is that in immunocompetent patients, there's mostly just a mild upper respiratory illness, but we're seeing some emerging challenges and infections in immunocompromised hosts. In the immunocompromised hosts in infants, we're seeing pertussis-like illness. However, it's a more mild presentation with fewer of the classic pertussis symptoms that are seen. There's shorter cough duration, and overall, there's a more mild respiratory illness when it comes to the respiratory presentation of infection. However, what we are seeing is that there is more commonly occurring with Bordetella Holmesii than we see with Bordetella pertussis, a systemic infection. And these infections in immunocompromised hosts, we see as bacteremia, cellulitis, and pneumonia. And this we very rarely, if ever, would see with Bordetella pertussis in immunocompetent or infant hosts. So we're seeing Bordetella holmesii as certainly an emerging and interesting infection that may warrant some additional attention in the future, as we're seeing increased numbers of immunocompromised patients, of course, in our population. However, bringing it back to Bordetella pertussis, we are seeing in the community that there is a resurgence of Bordetella pertussis following major vaccination strategies over the past 50 years. And there's two major factors that are the cause of this resurgence of Bordetella pertussis. One of the factors is waning immunity. And so we've initially with the Tdap acellular vaccine that had the Persistence toxin and various pertussis factors that were included in the vaccine. The immunity is expected to last initially about 30 years, but what research is now showing when we look at the serostatus of individuals is that the immunity is really lasting more in the range of 40 to 20 years, with demonstrated vaccine effectiveness in the younger age groups of 8 to 12-year-olds, 20 to 40%. So there's limited vaccine effectiveness, and in combination with a shortened immunity period, we're seeing that we have this issue of waning immunity. What also has been shown in animal models is that the initial acellular vaccine did not have a reduction in the nasal carriage of the organism, so there may have been additional transmission opportunities while an individual may not get the disease themselves. Without the reduction in the nasal carriage of the organism, you would potentially still be able to transmit the infection to other naive hosts. And so these factors together have impacted the immunity that's been achieved with the vaccine. However, in addition to those kind of factors addressed with immunity, what we've also seen is that there are molecular adaptations in the organism itself that are impacting the ability of pertussis to resurge in the population. So we're seeing molecular adaptations in the Persistence toxin, in Protactin, and in the Fimbriae that have been targets for the vaccine. And so that may allow those allelic variations to emerge in population. There's also been emergence of Protactin deficient strains, and this emergence began around 1994 worldwide. In fact, in 2012, the CDC surveyed the isolates that it had in its database and found that over 50% of the isolates tested by the CDC were in fact pertactin deficient. And this appears to be at least associated somewhat with this resurgence of Bordetella pertussis. But in combination with these things, we're also finding, and I think it's important to recognize, that around this time of recognition of waning immunity and emergence of molecular adaptations, we also have improved diagnostic methods, which is also helping us to detect more cases of pertussis. And perhaps this is also playing a factor in the resurgence of pertussis in the communities. So when we think about the diagnostic methods that we've had available to us over time, we find that there have been some challenges with diagnosis of pertussis. Culture for Bordetella pertussis is only about 60% sensitive, and the highest recovery rate for culture is with infants when the specimens are collected within the first 2 weeks of the cough. The challenge with this is that within those first 2 weeks of the cough, this can still be within this initial catarrhal phase where one may not have pertussis high on the differential because you haven't had that extended cough period, and it may just be perceived as an influenza-like illness. However, having cultures for Bordetella pertussis is important because it is useful to detect these mutations that can help us to understand how the vaccine is impacting the organism and allelic variants that may be emerging. And we can also use these isolates to detect emerging antimicrobial resistance. The CDC, in fact, has initiated some initiatives to recover more Bordetella pertussis isolates to combat these various factors. Also, there's specific media that are required for the detection of and recovery of Bordetella pertussis in the laboratory, and most laboratories don't routinely keep these media in stock. In the laboratory, the organism is quite fastidious and can take many days, a week or more for recovery on these specific media. So culture is just not a routine method that is done in the laboratory, though there are certainly some benefits for having culture available. Okay, so as we examine the options for diagnostic and the optimal timing for diagnostic testing for pertussis, we see that from the time of cough onset, culture is optimal only in those first 2 weeks. After that, and that's when pertussis really becomes more on the differential for clinicians, once patients have passed that initial catarrhal phase where the patient would have influenza-like illness and perhaps a more mild cough. After that second week is when the patient would begin to enter into some of the more classic pertussis symptoms. And that is when other methods become more available and are optimal for the diagnosis of pertussis, including utilization of PCR and molecular testing, as well as serological testing. So we'll look first at the use of serologic testing for pertussis. While it's not often used because it can be challenging and that it can require acute and convalescent specimens, the detection of the specific anti-Persistence toxin antibodies can be helpful and can be used as a sensitive way to establish infections when a significant duration of time has passed and relatively late in disease. But there are many factors that do impact the accuracy and the sensitivity of serodiagnosis of pertussis. This can include the time elapsed since the last vaccination, or if there was a previous infection, and whether or not only the primary series of vaccination has occurred, or booster vaccinations were given. In addition, the overall antigen content of the vaccine and the age of the patient are all factors that have to be weighed in when determining the clinical significance of the positive result. And so this is really a challenge when it comes to utilization of serologic testing and interpreting the result. Serology can't be used during the year following vaccination. And with all of these factors, there's really some challenges, and in some cases, limited diagnostic value for deciding treatment or prophylactic decisions for Bordetella pertussis. So this brings us to the utilization of molecular testing and molecular diagnostics for pertussis. When we're looking at this method, overall, we utilized insertion sequence elements as the primary target for Bordetella NAT assays. We utilize at least 3 of these insertion sequences to try to help us to differentiate between multiple species of pertussis or Bordetella, and then other factors can be used for further speciation. So looking first at the insertion sequence 481, it is present in Bordetella pertussis at anywhere from 50 to 200 copies. So this makes it a wonderful target for the detection of Bordetella pertussis. Because it's present in so many copies, we can get to a higher sensitivity in detection of Bordetella pertussis in clinical specimens. However, there is a challenge in that it can be present in Bordetella holmesii at a lower copy number of 10 to 50 copies. So this does bring up some challenges because any specimen that is positive for insertion sequence 481 could also potentially be not just Bordetella pertussis, it could actually also be Bordetella holmesii. Additionally, very few, only 3%, but certainly it's present in 3% of Bordetella bronchoseptica cluster 1 strains. But the insertion sequence 481 provides us with a good potential for use for the detection of Bordetella pertussis. Then we have the insertion sequence 1001, which is present in Bordetella parapertussis at a copy number of 22. But it is also present in 29% of Bordetella bronchoseptica cluster 1 strains, and a variant of this insertion sequence can also be detected in Bordetella holmesii at about 3 to 5 copies. The insertion sequence 1002 is present in Bordetella pertussis at a very low copy number, less than 10 copies, also seen at less than 10 copies for Bordetella parapertussis, and only in 1 copy for Bordetella bronchoseptica. So this is traditionally not used as a detection method for Bordetella species. Additionally, to help to increase the specificity for Bordetella pertussis, the Persistence toxin, which is only present in Bordetella pertussis, can be used to improve the specificity for molecular targets. However, it is only present in 1 copy, so we run into some sensitivity challenges. And then Rec-A can be used to help to differentiate those the detection of Bordetella holmesii, as it is present in only 1 copy for Bordetella holmesii and only present in Bordetella holmesii. So we see these multi-copy genes, and they're certainly helpful to support diagnostic sensitivity, which is needed when it comes to the detection of Bordetella pertussis. And this use of multi-copy genes is helpful because we can then detect at sensitivities less than or at 1 CFU. However, this does present a challenge because it could even detect colonization or carrier states. Using the insertion sequence 141, though, we do run into specificity risk because this insertion sequence is present in both Bordetella pertussis and in Bordetella holmesii. So how do we mitigate this specificity risk of the insertion sequence? One of the things that could be done is through review of additional sequences, such as the insertion sequence 1001, or to differentiate between Bordetella pertussis, holmesii, and parapertussis, but to differentiate between the presence of Bordetella pertussis and Bordetella holmesii, one could do periodic surveillance of Bordetella holmesii in your population to determine whether or not Bordetella holmesii is ever circulating in the population, or you could add a second target for increased sensitivity, such as the addition of the Persistence toxin or Rec-A to differentiate between Bordetella pertussis and Bordetella holmesii. So this could be, a potential model to look at. How would one determine whether or not you have Bordetella pertussis or Bordetella holmesii circulating within the population by the addition of Bordetella pertussis toxin detection and Rec-A to increase the specificity for Bordetella pertussis and Bordetella holmesii. However, we do risk at this point with the one copy gene sensitivity at low organism burdens that we often see in diagnostic specimens. So an additional method that one could use instead of the addition of the Pertussis toxin or Rec-A would be the addition of the insertion sequence 1002 to increase the specificity for differentiation of Bordetella pertussis and Bordetella holmesii because it is present in a higher copy number than Pertussis toxin and Rec-A. However, both of these present still the challenge of sensitivity and the potential loss of sensitivity while increasing specificity. So overall, it still, I think, becomes the best method to focus on the insertion sequence 1481 for detection of Bordetella pertussis, recognizing that there may be some carryover cross-reactivity to Bordetella holmesii, and doing periodic surveillance in the population to determine whether or not Bordetella holmesii is present in the community. In our laboratory, we use a laboratory-developed test identifying a primer pair that targets the insertion sequence 481, which would detect Bordetella pertussis, as well as some potential carryover cross-reactivity to Bordetella holmesii. We also have this multiplexed with the detection of Bordetella parapertussis using the insertion sequence 1001. This also includes an internal control primer pair that is used to monitor PCR amplification and or inhibition, and the assay is performed on the Liaison MDX instrument. We have evaluated in our laboratory two methods for detection of pertussis and parapertussis in specimens in the laboratory. We use one method and evaluated one method, which is an extraction method, where we extract 200 microliters of VTM from a nasopharyngeal specimen or nasal wash that is received in the laboratory. And then we utilize 5 microliters of the extracted nucleic acid in the master mix for detection of the specimen. We then were able to compare this method to a direct detection method where we take the nasopharyngeal swab that's in VTM and we utilize 3 microliters of that direct specimen of the VTM and add that to the master mix. In both of these cases, the samples were tested with the diasorin molecular primer pairs on the Liaison MDX instrument using the 96-well universal discs. It's important to note that these assays are laboratory-developed tests, and this was used with ASR reagents, and it's not cleared by the FDA. When we evaluated the performance of the direct versus the extracted method, we used 147 previously characterized specimens, and they were tested by both methods. We evaluated for the presence of pertussis and parapertussis among these specimens, and we find that there was a positive agreement of 92.9% and a negative agreement of 100%. Focusing on the 7 specimens that were positive by the extracted method but weren't detected by the direct method, we evaluated these discrepant results. And we looked at these results based on the CT range of the specimens. So what we can see is that when specimens were in the CT range of having a cycle threshold between 10 and 20, there was 100% agreement. And we see that same 100% agreement when we have a cycle threshold of 21 to 30. However, at the later cycle thresholds of greater than 31 is where we began to see the emergence of the discrepant samples. At 31 to 35, however, there were agreement with 20 of the specimens and only 4 discrepant samples. At 35 plus, we had 4 samples that are agreed, and there were 4 samples that were discrepant. And then when we look at the type of samples among these discrepants, we see that we had for Bordetella, Pertussis, and parapertussis, at the 31 to 35, there were 2 Bordetella pertussis samples that didn't agree, and 1 Bordetella parapertussis sample that didn't agree. The 4th sample among those 31 to 35 was an indeterminate sample that we weren't able to do further discrepant analysis on. For the 35 plus, cycle threshold value, we found 3 Bordetella pertussis samples that were discrepant and 1 Bordetella parapertussis sample that was discrepant. You can see for all of these that these have fairly late CT values in the specimens. So when we evaluate the analytic detection of pertussis, we determine the sensitivity of these 2 real-time PCR assays by measuring 10-fold serial dilutions of purified pertussis and parapertussis nucleic acid. We evaluated these at 10 to the 0 and 10 to the 7 genomic equivalents per reaction. And then we determined the average CT value based on the 5 replicates that were evaluated at each of the concentrations for each of the target performance. And what we're able to determine is that for the extracted method, we have earlier CT values compared to the direct method. They are slightly later. And then we, as we go down into increasingly dilute specimens, we find that by the time that we get to 10 to the 0 genomic equivalents, we're no longer able to accurately detect Bordetella parapertussis in the specimen for the direct extraction. We are able to still detect Bordetella parapertussis, but at a fairly late CT value. And so this does correspond with the data that we saw with our discrepant results, that we start to get later crossing thresholds and potentially begin to miss the detection of Bordetella pertussis and parapertussis once we get into the lower concentrations of organisms present in the sample as determined by this sensitivity study. So then when we compare the CT values for Bordetella pertussis and parapertussis for linearity in the extracted versus the non-extracted or direct specimens, we see that when we view our y-intercept, we do have a bias towards a later CT value for the non-extracted specimens. However, there is still a fairly good correlation of these CT values at 0.95 for the R squared, and the slope is at 1. But there is somewhat of a bias towards a later CT value, as we can see for the y-intercept. So what would be some of these causes as we look at the direct versus extracted values for pertussis? What would cause a late CT and basically a lower organism burden in the specimen? The timing from the specimen collection post-symptom development would result in fewer organisms present and a lower organism burden, as would the timing from specimen collection following antibiotic treatment. It's been demonstrated that patients with older age have a lower organism burden. And if there's environmental contamination with pertussis, that would also potential contamination sources, though they would be a false positive, they're usually present at a lower organism burden. So all of these would have an impact on the CT value. For the specimens and overall the organism burden that we would see in these specimens. So I'm going to shift gears just a little bit because we really need to kind of focus on what are we looking for when we're looking to detect pertussis in the clinical specimen. We can evaluate the analytical sensitivity of the test, but it's also important to understand clinically what are the specimens that we are receiving in the laboratory, because all of the factors that I've mentioned previously would all be influenced by these pre-analytical forces that would impact the specimen that we received in the lab. So just a bit about New Mexico pertussis epidemiology. New Mexico has experienced pertussis rates of epidemic proportions of pertussis since 2011. In 2012, New Mexico actually experienced a threefold increase in cases compared to 2011. And the U.S., even during that time, also experienced the highest incidence of pertussis since 1959. But historically, New Mexico itself has had higher pertussis rates than have been seen overall in the United States. When it comes to the overall vaccination recommendations, infants and children are expected to receive 5 doses of Tdap at 2, 4, 6, and then 15 to 18 months, and then once at a point between 4 to 6 years of age. Adolescents then receive a single dose of Tdap at 11 to 12 years of age, and then pregnant women are expected to receive a single dose of Tdap during every pregnancy at 12 to 36 weeks of gestation. So when we look more specifically at the New Mexico pertussis epidemiology and think about these vaccination recommendations, we see that the highest age group that most commonly has Bordetella pertussis, especially during these outbreak years of 2011 to about 2013, were children ages less than 1. So children in the infant age range were certainly an outlier. Patients ages at greater than 20 remained fairly low during this time period, but there is a slight bump during that outbreak set of years. And then the patients in the remainder of the age ranges appear to just cluster together in the rates of pertussis per 100,000 population. So when we look more specifically at the sources of infant transmission, we see that mothers at the age of 0 to 1 month of age are the most common cause of pertussis transmission, which is the cause and the need for mothers to get a vaccination during every pregnancy. However, it's important to note that siblings are the most common source of infant transmission for any age, from the 0 to 1 month of age or the 2 to 11 month of age. So it is certainly important to recognize siblings as a source of pertussis transmission for the infants in the population. So when we consider our pertussis cases and our immunization rates in New Mexico, what we see is that there really isn't a strong correlation between the numbers of pertussis cases and whether or not individuals are immunized for pertussis. We can see that the areas with the highest pertussis cases in the slide, in the image on the left, are in the darker shading. And the places with the highest rates of vaccination on the right side are in the darkish, are in the lighter shading. So the places where people are most fully immunized on the right side are the lighter shading. So we can see that there's really not a huge correlation that the places that have the highest cases of pertussis are not necessarily the places that have the lowest rates of immunization. So this shows us that we're in some ways fighting this uphill battle of effects of immunity and waning immunity and perhaps emergence of other strains of pertussis, as well as trying to control the number of cases. But this is also why surveillance for pertussis is extremely important. So when it comes to Department of Health needs, we in New Mexico utilize an enhanced pertussis surveillance because of the epidemic that we have been in over the past several years. And so we see the clinical case definition for pertussis, that in the absence of a more likely diagnosis, a cough illness lasting greater than or equal to 2 weeks, with one of the pertussis symptoms being the clinical case definition, and then a confirmed case is an acute cough with the isolation of Bordetella pertussis from a clinical specimen, or a PCR positive, or a contact with a case that has had contact with a laboratory confirmed case of pertussis. And so what we did in our laboratory was we entered into a partnership in collaboration with the New Mexico Department of Health, enhanced pertussis surveillance. We evaluated specimens with the Department of Health from 2013 to 2015. We evaluated 418 pertussis positive patients by PCR that also had a concurrent Department of Health investigation. Results from this investigation were aligned with PCR-CT values, and we correlated these results with the results of the Department of Health investigation. There were some limitations to this study. One, pertussis negative specimens in patients were not included in the study, and we did not have enough Bordetella parapertussis positive patients that also had an investigation for significance, and so they were not included in the study and in the evaluation. So when we look at the demographics of the patients who were included in this study to look at kind of the clinical factors that come into play with pertussis diagnosis, we see that there was a fairly even distribution between male and female. The age tended to be, the mean age was 14 years of age. When we look at the clinical data, we find that patients had an average of 23 days of cough duration, with a variety of presentation for the other pertussis, like the pertussis symptoms that would fall within the case definition. The most commonly occurring symptoms is the paroxysmal cough, but also present was post-tussive vomiting, whoop, apnea, cyanosis. We did not have any cases of acute encephalopathy or any deaths, but there were 35 or around 8% of the patients were hospitalized. There was only one case of 4 cases of a patient with generalized focal seizure. Interestingly, in only 72% of the cases were antibiotics given, and then 61% of the cases had ever received a vaccine. 11% of the cases were outbreak associated. When we look at the demographic profile of the patients to determine the percent of the population that met the criteria of a case clinical case definition for pertussis, we find that across the age range, around 80% or a bit over 80% of the population met the clinical criteria of the presence of the paroxysms of coughing or the other factors for pertussis case definition. Now, when we look at the CT value of the percent of the patients who met the pertussis diagnostic criteria, we find that the highest percentage of patients who meet the pertussis criteria had less than 30 CT value, so had a higher organism burden. So in the patients who had this higher organism burden, 90% of those patients met the clinical criteria, excluding the diagnosis of pertussis from that criteria, and only looking at the clinical factors. 90% of the patients with a CT value of less than 30 met these case definition. When you had the population of patients between 30 and 40, approximately 85% of those patients had the clinical symptoms associated with pertussis. And then of the patients who had greater than 40 CT value, this was a much lower number, but 70% of those patients had the clinical factors that would be associated with pertussis. Now, one of the things that we were able to do with our population is look at the impact of the symptoms and the demographic factors on the CT value. So what we did was we developed a reference patient that had kind of basic demographics. So we said this patient would be kind of our model patient that we would then compare every other demographic value against this patient. Gender had no impact on the CT value, as we saw earlier. So we just arbitrarily selected the patient to be female. We selected this kind of reference patient to be an adult, 18 to 64 years old, that they were hypothetically tested the day after, immediately after symptoms started, that they had never received antibiotics, and that we selected a fairly median CT value of 30. And so when we look at then the relative contribution of each of the demographic factors and then a couple of the more significant clinical symptoms, what we see is that, again, gender had no impact on the CT value, whether or not it would increase or decrease above or below 30. But what we do see is that in patients who are less than 1 year old, that significantly drops the CT value by 6.54 points. The other ages, with the exception of greater than 64 years of age, which also dropped the CT value by about 3 points. About 3 points. The remainder of the ages really did not have an impact on the overall CT value. When we look at the clinical presentation and the cough onset to testing per day, what we see is an increase in the CT value as we add each day of cough before testing occurs. So, for instance, if testing occurred the day after the cough occurred, there would be no impact. And then on day 2, there would be an increase of 0.12 on the CT value and a factor of 0.12 increase on each subsequent day of testing. And so this shows that the CT value increases as the cough duration continues. If patients received antibiotics prior to testing, that would add about 2.5 to our CT value. And so we can look at these factors and determine how the clinical factors associated with the disease and the demographic factors for the patient would play a role on whether or not we would be able to detect pertussis in a clinical specimen. Again, if we look at this data by age, we can look at the relative response to CT value and the symptoms by age. And what we find, again, is that in the patients that are less than 1, adding any of these clinical symptoms would decrease the CT value because there would be an overall higher burden. But in the older patients, we see that the CT value tends to go up a bit more because likely the patients are able to control a little bit better the infection that they may be seeing in the patient, and in the clinical specimen, we may have less of the specimen, less of the organism present in the specimen. So finally, in conclusion, what we see is that the changing epidemiology of Bordetella pertussis really highlights the need for effective and rapid molecular diagnostic testing. We certainly in New Mexico have seen an outbreak of Bordetella pertussis, multiple outbreaks in multiple settings, and we continue to have relative high pertussis rates here in New Mexico. So molecular testing that provides sensitive and rapid molecular diagnostics is certainly important. In our laboratory, we've used the Diasorin molecular Bordetella pertussis and parapertussis reagents, and these can be used to detect Bordetella pertussis and parapertussis with or without extraction. We find that lower CT values are positively correlated with a patient's likelihood of meeting pertussis case definitions and symptom presentations. And then finally, demographic and clinical profiles seem to have predictable effects on the CT value. And this can help us to understand a bit more about how sensitivity of certain tests and even the results in the lab can translate into a clinical response. So thank you very much for your time, and I'm happy to take any questions from the viewers of this presentation. Thank you.
Thank you, Dr. Culbreath, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. Our speaker will follow up with your questions via email. Okay, let's get started. Our first question is, are there Bordetella pertussis carriers who do not have symptoms but can spread to unvaccinated?
Definitely. I think as I was able to show in one of the slides earlier, the transmission to infants, many of those individuals, siblings, parents, grandparents, others, can certainly be asymptomatic, or they may just have some of them a more mild presentation of disease because they may be partially vaccinated, and so they can be carriers, and so certainly they can transmit Bordetella pertussis to unvaccinated individuals, and the most at risk for severe disease and in the unvaccinated population is most certainly infants. So it is certainly important for vaccination to occur in people who would potentially have contact with unvaccinated individuals, especially infants.
Okay. And how do you tell the difference between an active infection or a carrier when the CT value of the assay values of the assay are high, above a CT of 30? And do you have a cutoff that is informed by studies correlated with clinical symptoms?
So I think the question being, do we have a cutoff value that would distinguish between a carrier state or active disease? You know, that's really the challenge, is that a later CT value doesn't necessarily mean that a patient doesn't have active disease. It could still just be late in infection. We're talking about weeks of cough when it comes to the catarrhal phase and then even the convalescent stage. And then by the time an individual presents with pertussis, they're usually presenting several weeks after the cough has initiated. I believe our data showed the average cough duration at the time of testing was 14 days. So that's 2 weeks after the initial presentation of the cough. So there's this challenge with pertussis diagnosis. Do you have a cutoff where you decide that you're going to either no longer report or report some type of equivocal range where you say patient may not have active disease? I think that's a challenge. I think really it's important to understand the factors that impact a later CT value and to work in collaboration with clinicians and public health to help them understand that a patient who has been coughing for a long duration or a patient who may have been treated at some point would certainly have a positive result, but it may be later. I think it's important to help clinicians understand the mechanisms behind the testing, but we in our laboratory do not have a cutoff after 30 or after 35. If we have a positive result in our test, resulted as positive, and we do not utilize a cutoff because I think there's just some clinical challenges that make having a cutoff value even more difficult to interpret.
And how often are you monitoring for the presence of Bordetella homezii in your population? And what is the prevalence rate?
Yeah, so great. Thank you for bringing that up. I meant to mention that in the presentation. Among the specimens that we tested for the comparison between the direct and the extracted method, we also checked for Bordetella homezii, and we actually didn't find any in our population. So of just that about 147, I believe, specimens. So we routinely, we've done a couple of times over the past 2 years, is just done a couple of point prevalence studies where we'll take about 50 previously positive Bordetella pertussis specimens and run them to determine whether or not we have any Bordetella homezii. We haven't seen any in our population, but again, it's not a full surveillance study, but that's, I think, a great opportunity for continued collaborations with laboratories and their local departments of health to determine whether or not they have Bordetella homezii circulating in populations, and how the lab could help to support diagnosis and differentiation of Bordetella pertussis and Bordetella homezii.
All right, and we have time for one more question. In the last few months, what is the recent prevalence rate of Bordetella pertussis in New Mexico?
We unfortunately are still seeing fairly high rates of Bordetella pertussis, though they're starting to go back down. Right now, I believe the last data that we were seeing was around the five cases per 100,000. And so we're hoping that we can continue to see some decreases in those rates in our population.
All right, well, I would like to once again thank Dr. Culbreath for her presentation. I would also like to thank Dia Soren for making today's educational webcast possible. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon.